So there is a variety of different types of polymorphism ranging from single nucleotide change to very large scale changes. Okay, for evolution and speciation, such polymorphism play and very role important role. Okay, the technique of DNA fingerprinting was initially developed by Alex Jeffrey. Okay, he used a cellular DNA as probe that shows very high degree of polymorphism. Now that cellular DNA was also called as variable number of tandem repeats, that is VNTR. Now the technique involved southern blot hybridization using radio labeled VNTR as probe. Radio labeled VNTR as a probe. Okay, so it includes like isolation of DNA, then digestion of that DNA by restriction endonuclease, then separation of DNA fragments by electrophoresis, then transferring, that is blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, then hybridization using labeled VNTR probe, and then detection of that hybridized DNA fragments by autoradiography. Okay, now this VNTR belongs to a class of satellite DNA. So so the VNTR belongs to a class of satellite. VNTR belongs to a class of satellite DNA, referred to as the mini satellite. So VNTR is a mini satellite. Okay, it belongs to a class of satellite DNA. Okay, now a small DNA sequence is arranged randomly in many copy numbers. Okay, now the copy number varies from chromosome to chromosome in an individual. A small DNA sequence is arranged randomly in many copy numbers, right? The copy number varies from chromosome to chromosome in an individual. Now the numbers of repeats show very high degree of polymorphism. The numbers of repeats show very high degree of polymorphism. So as a result, the size of VNTR varies in size from 0.1 to 20 kb. So consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of different sizes. Okay, so these bands give a characteristic pattern for an individual DNA. Now it differs from individual to individual in a population, except in the case of monozygotic, that is identical twins. Now the sensitivity of the technique has been increased by the use of PCR, that is polymerase chain reaction. Okay, consequently DNA from a single cell is enough to perform DNA fingerprinting analysis. Okay, so it also uses in forensic science and all that, like, okay, and also in determining population and genetic diversities. So many different probes are used in generate DNA fingerprinting, right?